Hi all, Dr. Clark here again. Today, for this lab demo or lab information, I'm going to talk about procedure 3 of lab number 2. So remember, lab number 2 is biodiversity, and procedure 3 is introduction to biodiversity, the collection. Okay? So what you're going to do is you're going to do a biodiversity collection. If you were part of the live class, you would be doing a live collection. In other words, you would collect living organisms and then kill the living organism and present it as a biodiversity collection. That would either be insects or plants. Those are typically what we do. But as far as the internet sections or the virtual sections, you're going to do more of a virtual biodiversity collection. So instead of killing organisms, you're going to take pictures of them and present them that way. Okay. So let's dive into some of the nuts and bolts of this collection. It's worth a lot of points, worth 75 points of your grade. Okay. And it's an individual project, so each person in the course will do a biodiversity collection on their own. Okay. All right, so let's look at some things. First, you must have 20 living organisms. So you must take a picture, a high quality picture of 20 different living organisms. Okay, there are rules to these living organisms. They must be wild organisms. So there's no pictures of your pets, no pictures of things like indoor plants or garden plants or even outdoor shrubs and flower beds and things like that. None of those organisms count as wild. So you must have 20 wild organisms. Again, they could be fungus, they could be animal, they can be plant, and if you have the means to do so and to identify, you could even do protozoans. Okay? Please, even though I know bacteria and archaea are living organisms, please do not use bacteria or archaea. And the main reason for that is identifying it to species is very difficult and you're most likely going to be wrong. Unless you have capabilities of doing genetic analysis, you're not likely to get bacteria to species. Therefore, taking a picture of some bacteria growing on, say, a petri dish, that's not a living organism that's going to count for this collection. Okay, second piece, it must be in its native habitat. Now, that doesn't mean it has to be native to the region that you took a picture of the organism in, but it does mean that it has to be in kind of a wild place or a native habitat place, meaning no pictures of spiders in your hand or spiders in your house on your wall or other insects on your walls in your house or in a cage, anything like that. These have to be native habitat to the best of your capabilities. Okay, so like a plant on the side of the road is perfectly good for a native habitat. A plant in your gravel at your house in your driveway is not really native habitat. Okay, so taking a picture of something that gets established in an artificial region is not native habitat. The picture must be your picture. Okay? You have to be the photographer, and therefore there are no internet searches and pulling pictures off the internet, and no sharing of photos. So if Billy's got a good photo, you don't get to use his photo in your collection also. And I know that some of you might not be photographers, but again, the picture itself, we'll talk more about the details of the picture, but the picture itself, you don't have to be a photographer to take a good picture. If you take a picture from the internet, that is considered plagiarism, even if you give the individual who took the picture credit. You still plagiarize them. It's not your photo, it's not your organism, and you will fail the assignment. That is 0 out of 75 points if you take a picture from the internet, unless you're the author of the picture. So let's say you took a picture of a wild horse and you posted it on Facebook, and then you pull it off of your own Facebook account. You have all the details. Then, of course, that's your photo. You uploaded it on the internet, but you have the right to take it off. Okay, but you cannot use someone else's picture. And then the last one is they must be alive. So no pictures of roadkill or preserved plants or preserved animals or anything like that. They must be alive. Um, so hint 20 living organisms. Okay, so the requirements is it has to be a good photo, good quality. Again, like I said, you don't have to be a professional, but it needs to be high enough quality that I can identify the organism from the photo and just the photo. Okay? So let's say you go out and you take a picture of a butterfly, uh, swallowtail butterfly, and you knew that it was a swallowtail butterfly before you took the picture. Okay? You take the picture and you get in the picture you get a blurry head of the organism and nothing else. That's all you got in the picture. That's not good enough quality for me to identify it. Okay? You might have already known the identification because you were there, but you have to be able to identify the organism from the picture. So poor quality or unidentified organisms will result in -5 points for every single picture that the organism's not identifiable in. Next piece, you need the common name and scientific name. Okay. Then I need to know the exact location that you snapped that photo of at, and we record that in UTMs, and I'll show you an easy way to kind of get at that. So again, is exact location is going to be the GPS coordinates. It's not Rock Springs, Wyoming. That's not going to do it. Okay, the exact location, the GPS coordinates is needed. The date and the time, the weather. So was it sunny? What was the temperature? Was there any moisture? So was it raining, snowing? Okay. What's the wind speed? These kind of things. And then ecological habitat type. This is where you can need to kind of elaborate on what is the region or what is the habitat that this organism that you took this picture of this organism at. Okay, so were they in the shrub steppe? Were they in pine forest? Were they in the grasslands? Was it along a lake shore? These kind of things tell the individual who's examining the photograph more about the organism. What was that organism probably doing in that region? Okay? And that's the information you need for every photo. So I'll show you an example. Okay, so this is the example. I took this photograph of a mule deer, okay, Oticoleus hemionys. Okay, it was at this GPS coordinates, 13T614603, blah blah blah. Those are UTMs, and I'll show you the difference between UTMs and lat long in just a second. Okay. This was the date I took it, 7/24/2019 at 9:30 in the morning. Now, the key thing with the date has to be 2019. You have to have taken the photograph in 2019, okay? Or in 2020 if you're taking the course, in 2020 if you're taking the course in 2021, whatever it might be. It has to be the same year that you're taking the course. Now, it can be earlier in the year than when the class started, because I understand that in the fall, maybe maybe over the summer you took some really good photographs of some flowers or insects or mammals like a deer here, something like that, and you want to use those. That's completely fine as long as it's the same year um, that you're taking the course. Okay. Here you can see the weather. At the time, it was sunny, clear skies, 75 degrees Fahrenheit, 5-10 miles per hour wind out of the southwest. Okay. The photograph, here's the ecotype, okay, or the ecological region, the habitat region. This photograph was taken in Theodore Roosevelt National Park, which is in the Badlands of North Dakota. Okay. And the dominant feature there is sagebrush and tall grass prairie. And then I added a kind of note. There is multiple individuals in this region browsing on sagebrush and tall grasses. So it's in the region for feeding purposes, okay? And that's all there is to it. Okay. Then I'd go to the next one. Now, remember, it's the best way to set this up is on a PowerPoint presentation. So um, each slide of your PowerPoint presentation would be a new living organism. Okay? And so you'd have all the information for that living organism on each slide, and then the picture, and the picture has to be high enough quality. Now again, with the picture, you can see here in this picture, there's no doubt in most people's mind what the organism is that the photograph's about. Okay, so you can see that the deer is in the middle of the picture. Okay, it's the thing that's in focus. The trees in the background, which are perfectly fine for living organisms, and you could utilize this big leaf sage and this juniper. Okay, you could take pictures of them, but that's not the focus of this photograph. This organism is the focus of this photograph. It's easily seen that. Okay, so just make sure that you understand that when you're taking a picture, that you have the organism that is of interest as the main object within the picture. Okay? And so if I had like a little teeny bird over here, and you can only see maybe the back end of the bird, the rest, the head is out of the picture, and I say, oh, this is a raven. Right? Well, that's not doing the picture credit. That's not doing the organism credit, and that's not going to count. That's a poor quality picture because the organism was cut off. Okay. And one final piece. From the standpoint of photographs and identifying organisms and things like that. If you need help with identification, you can always send me the picture and a little bit of a description of kind of where the picture was taken at, what kind of habitat it was in, and I can help you identify it prior to the date that you're going to turn this in. This is due the very last day of class, and it'll be due through a Turn-It-In link, which you will upload your entire PowerPoint presentation. And that PowerPoint presentation will be of the 20 organisms that you virtually collected. All right, so I'm going to jump. Okay, so you can see that I'm showing you kind of how to use Google Earth, I guess. So Google Earth is a free program. Most of you know that. You can just type Google Earth Pro into the web browser and download the program. It's completely free. But it's very useful. So I know exactly where I took this picture, okay, of the mule deer. I know that it was in kind of the curve of this road, and the deer was here in this curve on this forested region. Because I know that, I can go to Google Earth and I can select Theodore Roosevelt, and I can jump over to the road and then see exactly pinpoint exactly where that organism was seen. Okay. Now, when you open Google Earth for the first time, if you are just downloading this, it will present your GPS coordinates in latitude and longitude. Okay, so if we jump over here, go to tools and you go to options, then you can see here show lat long, and it asks you how do you want to show lat long. Okay, do you want it, I think default is degrees, minutes, and seconds. Okay? But instead of degrees, minutes, and seconds, we want universal transverse Mercator, okay, or UTMs. Now, the reason why we do this is UTMs is universal across the globe. So let's say some of you take a vacation and you took a photograph. Maybe you're in Hawaii, or maybe you're in the Bahamas, or maybe you're in Panama. I don't know where you're going to go. Wherever you go, great. I'm glad you went. But let's say you took a picture there. By using UTMs, it's really easy. There are no negative numbers. You don't have to know which side of the equator you're on, or which side of midline of the planet that you are on. So there are no negatives, and UTMs just breaks the planet up into little plots. So there are no two spots on the planet that have the same UTMs. Okay? And that's why the uniqueness of UTMs is important. The other thing is it's much easier to use UTMs in programs like ArcGIS and mapping programs. So that's why I like students to use UTMs, because you'll see that if you progress into, say, computer science or something like that, you'll use UTMs a lot more often. Degrees, minutes, and seconds. Let's say you have a GPS device, okay, a Garmin or something along those lines, or maybe you use a GPS app on your smartphone. Most often it will present it as decimal degrees or degrees, minutes, seconds, something like that. If you don't know how to go in and switch it to UTMs, you can always write down the degrees, minutes, and seconds, and then you can just go to Google and say, convert degrees, minutes, seconds, or convert lat long into UTMs. And there's lots of converters. They're all free. And you can just plug in your decimal degrees and it'll spit out the UTMs. Okay, so pretty simple. You can see here that UTMs will be a number and then a letter, okay, that distinguishes kind of the region, and then it'll have an eastern heading and a northern heading, okay, and then the elevation is there also. So I would say Google Earth is really important, really easy for you to use, but utilizing it for this project would be fairly important and a good thing to do. There are other programs. If you like other programs and you can get the same information, great. Go ahead and use those programs. I don't really have a preference. Okay. All right. So with that, you can start collecting. And if you have questions about certain things, then let me know. Otherwise, I look forward to seeing all of your collections come the end of the semester.